Welcome, everybody. My name is Tommy Source. I'm Assistant Director of the Purdue Institute of Inflammation, Immunology, and Infectious Disease. And today on our Purdue Lecture Hall Series, I have the pleasure of welcoming Stephanie Oprescu, who is a PhD candidate out of Shihuan Kwang's lab in animal sciences. And Stephanie is really an expert in stem cells after all this work that she's been doing, if I may say so myself, Stephanie, but maybe you can tell us a lot more about what your real expertise is and your passion and how you got into research. And we have an audience here that is so interested in hearing your work on how do we make new muscles and regenerate muscles that have been injured and so on. So welcome, and thank you so much for taking time to provide this lecture for us today.
Great, thank you, everybody. So I will talk about my research, and then at the end, I can briefly discuss kind of how I got into research, since I guess the path was a little unconventional. First, I'll just talk a little bit about the work that I'm doing and sort of how we can understand a little bit more about muscle regeneration.
Awesome. Sounds good.
There we go. Is it working now? Okay, great. Okay. So in Dr. Kwang's lab, we study skeletal muscle, and why are we studying skeletal muscle? So many of you are probably familiar with what it is, but it makes up a really large portion of our body mass, so nearly 40%, and it's super important for maintaining your posture and kind of helps you interact with the world around you. And then, of course, it honestly makes you look really good. So it's kind of good to know how to build muscle and understand sort of what goes into the, building muscle and maintaining it. And the other important thing is that as you get older, having muscle, building muscle can really sort of improve your quality of life, and it can help with increasing bone density and recovery to different types of injuries. So in general, having muscle, maintaining muscle, super important. And overall, it's a pretty cool tissue. So I'm not sure how many people are familiar with the skeletal muscle anatomy, but I think it'll be beneficial to just go over it briefly. So there are over 600 skeletal muscles. I don't know can I use a pointer? How does that? It's okay. Okay. So there are over 600 skeletal muscles in the human body, and this is just kind of the structure here. So they attach to the bone via tendons, and they help kind of move your body in different planes. And this is just, a sort of labeled schematic of what the muscle is like. But basically, the entire muscle, so one muscle is tethered to the bone via a tendon. And then as you kind of go further into the structure, this entire muscle is made up of different fascicles. And each fascicle is made up of an individual muscle fiber or a muscle cell. So this is like a long muscle fiber, and it goes from each end of the bone. So it kind of tethers to each side of the bone, and that's one long muscle fiber cell. And so this muscle fiber cell is multinucleated, which means that it has multiple sort of nuclei in one entire long cell. Right? And so we can kind of, again, just a reminder of the sort of structure, we can look a little bit more closely at the structure in the lab, and I'll go over some of the details a little later, but just to be familiar with how it looks. So what we can do is we can take a cross section of the muscle. So we can take out a muscle and then cut it perpendicular to the direction of the muscle cells or the muscle fiber. And we can see here just the generic sort of structure of the muscle. You can see each individual circle is sort of a muscle cell or muscle fiber. And you can see that the entire muscle has a bunch of these different muscle fibers kind of bundled together. And again, you can kind of look more closely at it as well. So this would be just a zoomed in picture of sort of the structure. So you can see each individual muscle fiber has this sort of delineated green boundary. And so it's very clear. These are so this is like the individual muscle fiber cell. And so what's special about skeletal muscle? So to kind of study a little bit about what's special about skeletal muscle, we use a mouse model, and mice are pretty similar to mammals in general with respect to their skeletal muscle structure and sort of how they respond to environmental stimuli. So we can learn a lot from mice about our own biology and different mammalian biology. And the neat thing about the muscle is that we can sort of injure it. So we can take a muscle, so just in a mouse, and then we can inject some sort of physical agent, or in this case, this is a chemical agent. So it's cardiotoxin, which is super toxic. It's actually isolated, it's snake venom. So it will basically destroy your muscle and all of the cells and the muscle fibers that are in there. And we can easily injure the muscle, and then we can kind of analyze and look at the muscle and how it responds to that injury. And so this is what it kind of looks like. This is just, I don't know, I hope this isn't gross to anybody, but so this is the mouse you can see here, and I just kind of circled on the outside. This muscle is really accessible, so it's easy to inject a foreign agent into that specific muscle. And we can kind of peel back the skin, and you can see that the muscle is kind of tethered to the bone. So the bone here on the bottom, and then the kneecap. And it's the tibialis anterior muscle that's on the surface, which is sort of your muscle that runs along the front of your shin, is kind of the analogous muscle. And so we're able to inject it with this toxin, and then we can carefully cut it out. So we cut it from the tendon, from one end of the tendon to the kneecap to the other end of the tendon, and you can see it's pretty clearly sort of isolated. And then we can do different analyses on this muscle. And so again, this is just kind of a schematic of like what we're looking at, how we look at things, and just a reminder. So when we're looking at the cross sections in general of the muscle, it's just an entire cross section of the muscle. So it'll represent this entire sort of bundle of all these different muscle fibers. And so in normal muscle, you haven't injected any foreign agent into it, you haven't injured it. You can see that the fiber boundary, sort of here in this case in white, are pretty clearly delineated. Everything looks pretty clean. But then immediately after, in this case 3 days after you inject this toxin, you can see that there's a lot of damage. So a lot of these really nicely and really clearly delineated fibers are kind of ruined, and the little purple dots in this case are nuclei. So those kind of can help us demarcate individual cells. And those, you can see there's a ton more of those than there is in the non-injured. But slowly, as you sort of progress and the muscle can continue to regenerate, you can see that there's a lot less of these sort of infiltrating different cells. So these nuclei in there, you can see that all of the muscle fiber boundaries are starting to look a little bit better. And one of the sort of markers of a muscle that's been injured and has been repaired is this sort of central nuclei. So it's basically a dot in the middle of each individual fiber. So that indicates to us that this muscle fiber was injured and now it's been repaired. And by 21 days, so later after injury, this is in the mouse, it's a different timeline in the human, it looks pretty similar to the non-injured muscle, with the exception of this central sort of nucleus that suggests, or that tells us, that this was once a damaged fiber that's been repaired. And so kind of what gives muscle the ability to be able to get injured so brutally and then more or less fully repair and restore normal function? And I think we'll just do like a brief background on sort of stem cells, and then we can talk about the unique stem cells that are present in the skeletal muscle. And so maybe some of you are familiar with, in general, stem cells are pretty a hot topic, I guess, these days, and their ability to help you repair damaged tissue or help you look younger. But a lot of these stem cells come from one of the original pools. There's the human embryonic stem cells. So human embryonic stem cells are kind of develop early during, your gestation, or when you're starting to develop as an embryo. And so they come from the inner cell mass, and these cells can basically divide a bunch of times, which is termed self-renewal. So they can continue to expand, and generate a bunch of different clones or copies of like one another, right? And the neat thing about human embryonic stem cells, or embryonic stem cells in general, is that they can give rise to all of the tissues in your body. So this is called gastrulation, and essentially these in response to different cues during development, they can become either neural cells, skin cells, muscle cells. They give rise to more or less all of like the tissue in your body. And these are termed pluripotent stem cells. But there are a lot of types of stem cells, and so kind of define these types of stem cells and then move into sort of what is special about the muscle. And so totipotent stem cells, these are even, more potent than the human embryonic stem cells. So that can give rise to an entire organism. And an example of that is a fertilized egg. So that basically gives rise to everything you need, all of your tissues and all of the structures that are needed in order to support the developing embryo. And then we have the pluripotent ones, which we just discussed. And so those are the human embryonic stem cells, and they can give rise to all of the different germ layers, all of the different tissues in your body, but they can't give rise to a fully functional organism on their own. And so the example for those are human embryonic stem cells, which we talked about. And then there are multipotent stem cells. So these stem cells kind of have a limited range, but it's still a pretty large range. And an example would be mesenchymal stem cells. So those stem cells can give rise to maybe a fat tissue or a bone cell, so an osteoblast. They can give rise to very limited, but also kind of diverse pool of different cell types. And then more restricted than that is an oligopotent stem cell. So those only give rise to cells that are pretty similar to one another. And an example of this would be hematopoietic stem cells. So these can give rise to different cells of the immune system, but they can't give rise to, say, like a neuronal cell, because that's kind of too distant. And then we have these unipotent stem cells. So those can only become a specific cell type. And the example for this is a muscle stem cell. And so muscle stem cells are really kind of what give the muscle a lot of its ability to sort of respond to injury, regenerate, and be a totally normal tissue again, right? And so again, this is just unipotent muscle stem cells. So we're just a little bit about the structure again, just to kind of remember where we're at. So this is your entire muscle. Again, the muscle is made up of all these bundles of muscle fibers. These muscle fibers actually, each muscle fiber has a unipotent muscle stem cell, well, 1 to 10, say, that kind of reside right along the muscle fiber. And so this is sort of what it looks like. So you have your individual muscle fiber, which is the muscle cell. It has multiple nuclei, and that's really the structure we talked about over on the left. And then the muscle stem cells will stay really close to that muscle fiber, kind of waiting for any sort of damage or any sort of signal that might suggest that there's something that they need to sort of respond to. And so what does unipotent really mean if they can only give rise to one sort of cell type? Why is special, and what is sort of unique about this? And so usually the muscle stem cell is sort of in a quiescent state. So it's quiet, nothing is really happening. You haven't injured the muscle, there's no damage, everything is kind of normal in the tissue. But if you injure the muscle, either say you cut it or you inject, in this case, the foreign agent, the cardiotoxin, which is the snake venom, the muscle stem cells will sense this injury and they'll start to proliferate. So they'll generate a bunch more of these muscle stem cells. And so the pool will go from 10 to maybe 100. And then a subset of these muscle stem cells will actually start to differentiate. So they'll kind of move along in order to repair the injured muscle fiber. But interesting is that a subset of these will actually go back and sort of maintain that original pool of muscle stem cells. So you have this balance in between sort of cells that will help to repair and then cells that will help to maintain the original pool of muscle stem cells. And so then these sort of post-mitotic muscle stem cells will kind of fuse together and they'll repair the injured muscle fiber. And so that's just delineated here. You can see these central nuclei, which is sort of the characteristic feature of a muscle fiber that's been damaged. And this is the broad sort of trajectory that these muscle stem cells take when you injure the fiber, the muscle fiber. Right, and so as we briefly discussed, this is again just recapitulated here. You have these sort of muscle stem cells, they're just hanging out, not really doing too much. And then they sense the damaged fiber that you've injured, and then they sort of kind of come to help activate, proliferate, help and repair this sort of damaged fiber. And then you have, again, a fully restored functional muscle. Right? Okay. And so how can we sort of visualize these muscle stem cells? So there's a lot of ways we can visualize them, but I'll just go through briefly a little bit of how we do this. And so one of the ways is using immunofluorescence. So basically what we're able to do is we know sort of proteins that are expressed by the muscle stem cell specifically, and then we know proteins that are expressed by the muscle fiber. And so we can use a technique, I won't go into too much detail about this, where we're able to actually visualize these proteins and see how really they are specific. So you can clearly see here the in green is the muscle fiber boundary, and you can see how the muscle stem cell, in this case in pink, is right next to the muscle fiber, right, in both of these images. And so in blue is a nucleus. So these are either nuclei of cells that are in the muscle, or these are the myonuclei themselves, so the nuclei of the actual muscle fiber itself. And then another way that we can look at the muscle stem cells is we can actually very carefully cut out this entire muscle, so from tendon to tendon, and do a series of digestions in order to release these individual muscle fibers. And so muscle stem cell, muscle satellite cell are somewhat interchangeable names. So this is a muscle stem cell. And so kind of in white along here, we've just demarcated the outline of the muscle fiber. And so if you look at the middle panel, you can see that the muscle fiber has a ton of these different nuclei sort of aligned along it. And we can see that along this individual muscle fiber that's pictured over here, that there are two in this frame muscle stem cells or muscle satellite cells that are actually still associated with the muscle fiber. Just the overlay of the nuclei with the muscle stem cell staining. And so, okay. And so we can do this again, this type of visualization, at least with the cross section, where we can look at the muscle. Okay, so again, we've injured the muscle, we're taking a cross section, so we're cutting perpendicular to all of those muscle fibers. And so this is what a normal muscle looks like, no injury, everything is okay. There may be 3 like pink dots here, so these are the muscle satellite cells or the muscle stem cells. And you can see that there are not very many of them, but there are some. And then, yep, that's just, shown right here. And so then around 5 days after injury, when a lot of the injuries happened, you can see that there's a lot of sort of damage, a lot of empty space here. And then all of these fibers here that are delineated in green have these central nuclei, which indicates that they've been repaired. And there are a lot more of these pink dots in this frame that we're looking at here because the muscle stem cells have been actively responding to the injury to help repair it. And so you can see kind of pointed here, again, the muscle stem cells are in the tissue, but there's more of them as they're responding to the injury. And then at 10 days after injury, again, so this is sort of progressing along. So this is sort of the immediate damage where it's really obvious. Now it's starting to look a little bit more like the non-injured muscle. Again, you can see the muscle stem cells here. And you can see that the muscle fibers all have these central nuclei, meaning that they've been repaired. So they were injured and they were repaired by the muscle satellite cell or the muscle stem cell. But there's not as much sort of empty space here. So it's really kind of functionally starting to resemble the non-injured. And then by 24 days after injury, you can see it's pretty much repaired, sort of relic that's of that centrally located nuclei. And you can see again the muscle stem cell, you can see it's there. So a subset of these muscle stem cells that have, helped to repair the damaged fiber are already sort of differentiated and fused into these injured fibers. And then another subset of these have actually kind of stayed in order to maintain the pool in case there is any other injury again, and they need to respond again and do the same thing. So in blue is just nuclei, which again mark individual cells. And so we can see the muscle stem cell, great. But there are a bunch of other nuclei. I'm not sure if anybody appreciated that, but there's a ton of other nuclei in this sort of, open area here, and then near the fibers, there's a ton of other cells basically that are also actively engaged when the injury happens. And so what are these other non-muscle stem cells that kind of help for repairing the muscle? And so again, this is sort of the quiescent states, the muscle's quiet, nothing bad has happened yet. And you can see the muscle stem cells just hanging out along the muscle fiber. And then immediately after injury, you have this damage, and this damage is sensed by the muscle cells. So they start to kind of proliferate, activate, and sort of kind of start to undergo the process that they need to in order to repair the damage. But also all of this sort of debris that's caused by these dying muscle fibers needs to be cleaned up. And so we have different immune cells such as macrophages that will come in to help clean up that debris. But kind of it's important that once all of that debris has been cleaned up, that those macrophages themselves are not still trying to clean up things, because that can actually cause more damage long term. So there's sort of this transition of this response where the macrophages that are eating all of the debris and getting rid of it kind of stop doing that and transition in order to help the environment become more sustainable for the actual repair of the muscle. And then eventually the fully repaired, fully functional, more or less like the quiescent, non-injured muscle is kind of seen here, again, with the central nuclei. So there are a lot of other cells that are involved in this repair process. The muscle stem cells really take a lot of the attention, but without all of these other cells, the repair also wouldn't happen. So how can we really identify other cell types that are involved? And so I'm sure that many of you are familiar with the basic sort of structure of a cell. So every cell has a nucleus, and in that nucleus, it has DNA. That DNA is transcribed into mRNA, and then the mRNA is translated into proteins or polypeptides. So every single cell in our bodies has the same DNA. So how can we kind of distinguish all of the different cell types that are involved? Well, they have different RNA, and they also have different protein. And so this is kind of just a picture here. So I guess in this case, you have the cell that has purple RNA and purple protein. So that's defined sort of by this class of purple RNA and purple protein. And then the same with the green RNA, green protein. So all of the same DNA, but different mRNA, different protein is expressed in a way that is so unique that we're able to sort of distinguish maybe what a macrophage is versus a muscle stem cell based on the RNA expression. And so this has been a really powerful sort of base of knowledge for us to kind of explore what cell types are involved in repairing the muscle. And what we can do is we can dissect all of the tissue. So from, let's say, like an injured muscle, dissect all the tissue, release all of these cells so that they're kind of not, clumped together anymore, and they're kind of individual cells. So all of these sort of individual cells. And then we can actually get the RNA from every single individual cell. And that can kind of help us see, like, okay, which cells are involved in muscle repair. And so the way we do this is using a technique called single cell RNA sequencing. And I'll try and keep it, it's a neat technique, it's fun. So essentially you have this heterogeneous tissue. So this tissue here, it has a bunch of different cell types. And if you were to just isolate all the RNA from the entire tissue, you would have no idea which RNA came from which cell type. But we can use single cell RNA sequencing. So basically what we're able to do is we can take all of these cells individually, and we can actually get a unique RNA profile for each one of these cells. Oops. And so every single dot in this case represents an individual cell that has a unique RNA profile that can help us identify what type of cell it is. And so through a series of analyses, you can kind of take all of these individual cells, which we have an RNA profile associated with, and then we can actually group them together on a plot. And we can see, okay, so these cells in red, these all have a really similar RNA profile. Not the exact same, but really similar. And then the same we can do with the different cell types. So this will kind of like cluster our cells or group them based on how similar their RNA profiles are, while still retaining all of the information for every single individual cell. So all of the RNA that's expressed in every single cell. And so this technology is a little fancy. I mean, I guess it's fancy, but it's pretty streamlined at this point. So what we can kind of do is we don't have to actually isolate RNA from every single individual cell, because that would be extremely tedious and probably a lot of room for error if we were trying to get RNA from every single cell. And so this really neat technology came out where we can kind of use oil droplets in order to basically we pipe all of these cells in. So each individual cell gets kind of piped into an individual oil droplets with all of the necessary components we need in order to get the RNA profile from that individual cell. So in one oil droplet is one single cell plus all of the things we need to isolate the RNA. And then we can basically pool everything together because it all stays separate since it's oil. And so then we can do a lot of this analysis in bulk. So in each individual sort of oil droplet, there's a barcode, and that barcode is going to help us identify all of the information that came from one cell. And so we kind of do all of this in bulk, which makes it a lot easier. And then we can do a lot of the analyses in order to sort of identify the cells that are involved. And so this sounds really complicated, but it's really not. Basically, there's this fancy sort of company, thankfully, that has developed this sort of small box, which is a little anticlimactic for how fancy this sounds. But you basically load all the things you need into this box, and it more or less does this for you. So it's fun stuff, and you get a ton of information, right? And so this has kind of helped us to define and understand a lot of the cells that are involved in muscle repair. That's not just the muscle stem cells. And so what we did, so again, we can injure the muscle, and we have this, again, non-injured muscle here. We collected all of the cells from the non-injured muscle. So we collected the individual cells from non-injured. We also took some cells from earlier time points after injury. So you can see there's a lot of damage here. This is when a lot of the debris is being cleared up. A lot of those macrophages should be present that are helping kind of clean up the environment. And then sort of later on, when a lot of these muscle stem cells are starting to participate more and they're starting to repair the injured fiber, and eventually you have almost complete repair through to when it's essentially fully repaired. And so we were able to take cells from all of these different time points and kind of get an idea of what the RNA profile and what cell types are involved at all of these different time points. And so if we remember here, this is just all of the tissue. So from all of the time points, right, we have all these tissues, all of the individual dots. Each one is an individual cell with a unique RNA profile. And so we can do some analysis, and then it'll allow us to kind of group cells together based on how similar their RNA profiles are and to help us define cell types. And so this is sort of what the data looks like from all of the injured muscle. So it's from every time point. Basically, I think it might be a little hard to see, but each individual dot, if you wanted to, you could go in and you could look at the RNA profile that's expressed in this individual dot and get a ton of information about that one individual cell. And so this is over 50,000 cells that we were able to analyze and collect. And they kind of already naturally, with the analysis, will group together based on how similar they are. And so, for example, the biggest population that we got is this immune cell population. And there's a ton of them. I think it's over 20,000 immune cells that we were able to collect from all of the different time points after injury. And we can look a little bit more closely at the immune cells, which I think just nicely sort of demonstrates how this technique works and also how nice and progressive muscle repair actually is. And so that's just, we're going to just look at this population here briefly. And so the non-injured muscle, so we haven't injected anything into the muscle, it's just resting muscle. We take it out in color, it are individual dots, and these dots are individual cells we collected from the non-injured muscle. There's basically no immune cells, really very few that are present in the non-injured state, homeostatic state. But then immediately at the early time points after injury, we can see that there's this population here, sort of progressively kind of moves along in a clockwise trajectory. And so this immediate response is likely cells that have infiltrated the muscle because they've sensed the damage. And so they're helping to clean up the debris. And then these cells, either cells, the same cells, or maybe more new infiltrating cells, we can't really delineate that with this technology, will actually sort of transition. So their RNA profiles slowly become a little bit different. And as they progress, as they go from sort of this RNA profile that is indicative of them cleaning up the debris and helping to repair, sort of like getting rid of the damage. And then as they sort of start to transition towards helping actually provide an environment that's more favorable to repairing the muscle and not just cleaning up all of the damage. And this sort of continues at the later days. So you can really see these cells are just kind of progressively becoming a little bit different, right? As you kind of progress through the repair. And so later stages, again, they continue sort of counterclockwise. And then again, continuing here with less of them at the later time points, because there's not as much damage at this point, the muscle's almost repaired. And then at the really sort of later stages where it's almost fully repaired, more or less repaired, you can see that there are way fewer immune cells than at these early time points. And it's kind of returning to this homeostatic state, but there's still going to be some immune cells there in case there is any sort of other injury or anything that they need to respond to. And so this is just kind of highlighting really, I think, how progressive this response is and how much information we can really gather from this single cell RNA sequencing, right? And so this is a very fun technique, I think, for me to learn in the lab and to analyze all of the data. And the amount of information is really incredible when you think about just one dot being one cell that has a ton of information on it. And so it's a fun time, I think. And so really what we were kind of able to get is sort of cells from all of these time points, from the immediate response to injury all the way through to repair. And this is a really kind of loud picture, I think, which I don't love, but I think it kind of highlights how it's a progressive sort of response. So immediately once you have this injury, you have the degeneration of these muscle fibers, immune cells that come in and that help to clean up the debris and really lead to this really inflammatory sort of environment, which is favorable to cleaning up the debris, but eventually that kind of needs to transition in order to help the muscle stem cells actually repair the injured fiber. And then eventually those repaired injured fibers, they're originally a little bit smaller, but then they get bigger and bigger as they sort of mature and almost, kind of repair fully to being fully repaired, with the only relic that's really left being those kind of central nuclei where at one point at some time this muscle fiber was damaged. And so kind of what's next? So there's a lot that we're doing, in the lab, but what this really kind of helps us understand is what proteins might be expressed, for example. And so if we know all of the RNA profiles, the next thing is, okay, so does this RNA actually get translated into a protein? And so from that, we can kind of understand a little bit more and generate hypotheses about maybe how these cells are actually physically interacting with one another. And so this is a little bit of a maybe busy figure, but the takeaway here is that we have, in this case, a T cell and an antigen presenting cell. And so we know that these two cells will interact with one another in the context of the muscle. So but what does that interaction actually lead to, and how maybe might this interaction help the inflammatory sort of macrophages or antigen presenting cells become less inflammatory in order to support the regeneration. And then we can also look into how the muscle stem cells themselves are actively engaging with the immune cells that come in. So a lot of the work now that we're doing in the lab is looking at how muscle stem cells might be interacting with some T cell populations to see maybe they're actually influencing the behavior of these immune cells in order to help generate an environment that's more supportive for their function once all of the debris has been cleaned up. And so kind of course, like what's next? Another thing too, I think that's really important with this is we're kind of understanding maybe if you have a lot of inflammation in the muscle, if you bring in a different cell type, those might uniquely interact with some other cell type and say make it less inflammatory, and that can kind of help resolve some of the damage. And we know that in different muscle disorders such as Duchenne's muscular dystrophy, there is sort of a lot of this inflammation and damage, continuous damage of the muscle. So is there any way that we can think about some of the cell types we identified and how they might be interacting with the environment to see if that can actually reduce some of this inflammation and degeneration that we see in some of these dystrophic diseases, and with the hopes of eventually kind of repairing the muscle and leading to sort of helping people with having a normal life. And so that's, I mean, that's really the big takeaway, I think, from, let's see, from our lab and what we're working on. And so this is just an image of the lab that, you know, we all took. I think this was what, maybe like last summer. So obviously huge thanks to my mentor, Dr. Kwong, and then two undergraduates that I worked with, and Anna and Nick, and then my big thanks to my thesis committee and some help from the Olson lab as well. And so that's really the like what we're working on in the lab. I actually, so when I ended up coming to Purdue, I never anticipated that I would do research. In high school, I liked biology, but I think sometimes it can be overwhelming because there is so much memorization and things that you have to learn. But I also just want to reiterate that I promise, like, the more that you learn and get that foundation, the more exciting and sort of like imaginative you can be in biology. And so there's so many things to discover, and like the textbooks maybe make it seem like we know everything, but we really don't know that much. And so like it's truly so much fun. And if there's any creative urge in you, I actually think research in the biological sciences is a really exceptional sort of path to go down. And I studied Spanish in undergrad, and then I was really lucky and ended up sort of taking a genetics class, having some time in the lab, and then seeing what lab work is actually like and falling in love with it. And it's kind of been a love affair ever since. And so get lab experience and just like let yourself be creative too, because that's a lot of biology as well, and think outside the box. And some idea may sound crazy, but it's probably not. So just, you know, biology is fun. It's really fun, I promise. So yeah.
I love the way you put it. And it's so amazing. So you first started into, like, you were going to do Spanish. Spanish was going to be your major, is that right?
Yeah, so my major is Spanish, actually. Yeah.
It still is your major is Spanish.
Yeah.
Okay.
Yeah.
And then like you just got exposed to research by volunteering in labs?
So I was like considering pre-med. I wasn't really sure with a Spanish major, and I accidentally ended up registering for like a graduate human genetics course. And that human genetics course was really engaging with the primary literature and learning how to develop an experiment. And that completely changed my frame of thinking about biology. And so I got in touch with the professor who directed the course, and he was like, I have a spot in my lab if you want to try research. And then that was it. Yeah.
Awesome.
Yeah.
Awesome.
It's really fun, everybody. Really, it is.
So I mean, what advice would you give a high schooler who really they don't know what they want to do yet? And I mean, it sounds like, you know, the adventure is open.
Yeah, don't close anything out. I think, you know, it's it's easy to have an idea of what you want to do, you think maybe in high school, but just stay open because that can really change and you never know where you might end up. So just keep an open mind is really my big advice. Yeah.
What about the courage that it took to say, like, okay, Spanish, great, but I really like that. Should I jump? Like, how prepared were you with the biology and the math and all the preliminary courses that you needed to take if you had already sought direction in the languages?
Right. So I was kind of fortunate in that I was like continuously considering pre-med. So I was taking a lot of those courses along the way as I was doing my major in Spanish. So I actually have a bachelor's of science with a major in Spanish.
Yeah.
But I will say that like one of the things I think, at least with finding what you're passionate about, is that it never feels like courage. It just feels like something you're excited about. And so just if you if you can find that thing, then just trust yourself and let yourself love it.
Yes,
Yeah,
that's awesome. Awesome. And Stephanie, like you, many of us are passionate about many things, right? Interested in so many different things, languages and arts and sciences and life, right?
yeah, exactly. Yeah, it doesn't, I mean, of course it doesn't have to, it can, yeah, yeah. I'm very fortunate to have found this, I think, but I think that it's in everybody. I really do.
I
Yeah.
like that. You seem to have worked really hard, though, to get beautiful slides together for us today and beautiful data. It seemed like with all the amount of data, that you have to be pretty savvy at handling that data, computational, strategies and things, databases. And how's that been? Has that been enriching? Has that been a big challenge, or how do you manage that?
Yeah, so it's been a challenge, but I think when you have data that you generate like that yourself, like you very much want to explore it. So figuring out how to analyze all of the data is kind of like, okay, I need to do this because I'm really excited to see what's in the data. And unfortunately, I hate to say this, but Google is your best friend. So a lot of the stuff I learned probably through watching YouTube videos and getting in touch with other experts, but it's a lot of trial and error too. So there is a lot of ups and downs. It's never, if you're flat, then like something's not right. Like you better be really excited and then really frustrated. And so that's, yeah.
I
Yeah.
like the way you put that. Because it can be hair pulling, right? Like so
Oh yeah.
frustrating, things that, oh my gosh. And then just the amount of data, it just seems like it's a lot. I want everybody here to understand that the sequencing technologies are getting better and better and better. And that means that the sequence data that we're acquiring, as you put it, Stephanie, not only DNA sequences, but now we can sequence RNA and understand what genes are being expressed by looking at the profile of RNA genes that are the RNA sequences that we can capture. Tell us a little bit about these new technologies coming about in sequencing.
Yeah. So, I mean, they're pretty incredible. I mean, now I think single cell sequencing, what I briefly talked about here, more or less everybody is doing it. So you can get an idea of every single cell that's present in a tissue that you take and what that RNA looks like. And if there's this sort of a disease state or something, you can see like, oh, what's wrong with the cells and what type of genes are these cells expressing under a disease state versus not. And you can get, you from one individual cell, you can see maybe over 10,000 different RNAs that are expressed. And so that gives you such a deep picture of the state of the cell itself and what it's doing. And it can give you a lot of information about what it's doing, how it's reacting to the environment, and in general, like what is its identity. And I think that's really it's a lot of information, but it's really, really great to have. And you can ask a lot of questions and find sort of hypotheses from all of these data.
And I really like also the way you put it, that, you know, don't be hesitant to jump into the challenge and gain some of the knowledge, some of the techniques, so that then you can be creative and ask these questions. Are we anywhere close to answering any questions? Most of these questions, I mean?
I think you answer one question, you open up a whole box of another set of questions. So, yeah, we're getting there. Yeah, sure. Yeah.
That's awesome. That's so great. So it's really technology that is exploding. It's really helping us understand a lot more about the dynamics of expression of genes and what gives the characteristic of these different cells.
Yeah, absolutely. And I think like one thing we're working on now is we found that the muscle stem cells themselves can actually express some sort of inflammatory genes. So genes that are not normally, we didn't really know that these muscle stem cells could express them. And so we're able to kind of work off of that to understand, hey, wait, like everybody thinks that the immune cells come in and they sort of affect the muscle stem cells in the environment, but nobody really has looked at the converse of how are these muscle stem cells actually affecting the immune cells that come in and sort of helping the environment as well. And so this single cell sequencing has kind of opened up this area of research in the lab that we're working on now to think more about like a muscle satellite cell actively engaging with its environment as well, rather than just kind of being passive and just, fusing together, repairing the fiber, and all the other cells doing the important stuff in a way. So that's, yeah.
And so there's a lot of community help, you would say, from the other supporting cells that are around the area.
Oh yeah, without them, the regeneration also would not happen. So
yeah. Thank you so much for taking us through that. And I thought it was great, the way that you explain all the different types of stem cells and their limitations and the different origins of them and so on. I think that was really great, and it was something that I learned a lot about today from hearing from you, Stephanie.
I hope it was informative to everybody, and get into the lab. That's the thing.
Get into the lab, take the courage. This is something that we say like a broken record, but it really does take that courage and people taking the leap and asking questions like you did, right? Taking the courage to ask the questions.
Yeah. That's the other thing I want to say. Like I know everybody always says there's no such thing as a stupid question, but I promise there is no such thing as a stupid question, because it gets you one step closer to being able to articulate questions that will be really important. So just keep asking questions all the time, just all the time.
All the time.
Yeah.
And then what about being satisfied with your own conclusions? Do you like that?
Being satisfied with your own conclusions.
Yes. So like, should you always think that you're right with your own conclusions?
Absolutely not. I think that's one of the things too, is always be ready to scrutinize your conclusion as well. So go back and ask that fundamental question again, right, of your own conclusion, and really kind of put it through the ringer. Just never stop asking questions. Really.
That's right.
Yeah.
So, I love it. I love it because I mean, even you as an example, right? Your first conclusion of Spanish being the way to go was something you had to go back and you had to question. Asking people, talking to people and asking them questions. I think those those tidbits of advice that you're providing are so important for people to hear. And thank you. Thank you so much for doing this. And thank you, everybody, for joining in today.
Everyone.
Stephanie Oprescu, wishing you all the best as you
Thank
finish out your PhD and you continue to move on to your career.
Yep, here we go. So thank you,
Awesome.
everyone.
Well, thank you, Stephanie. Take care.